Hi, everyone. Dr. B here again. Thanks for joining us for another episode of Ask the Dentist. Today has a video portion to it, and I encourage you to listen and watch the video portion. Certainly the audio portion is interesting enough, but today we are very lucky to have, again, Dr. David Lin, PhD, the Chief Scientific Officer at Bristle, and we're going to be talking about how to interpret, why we should test our oral microbiome, and then how to interpret the results. A lot of you have tested, and I think that's wonderful. I think that's great that you're interested and that you're doing that. You're going to get a lot of information. You're going to save yourself a lot of money and a lot of time, and you're going to learn a lot about your oral health, and not only that, but your systemic health. Your predisposition to Alzheimer's, to gum disease as you get older. A lot of great information just by spitting into a little vial. But today we want to focus on what to do with the results. That's always the crux. We've got a great test. We have the first high resolution shotgun kind of testing of all 700 plus bacteria in the mouth and more, and Bristle provides that. But what do you do with that information, and how does it convert into actionable actions behaviorally, but also what products do you use? There's so many things in our environment and in our lifestyle. Diet is another one, environmental pollutants, bad dentistry, toxic dentistry, even fluoride. There's some new research on fluoride and its effects on biomes in animals and in humans as well. What's wrong with our oral microbiome, and what can we do? What can we eliminate? What can we add? How can we fix the oral microbiome? So again, David, thanks for joining us today. I see you as many do, one of the experts in the oral microbiome. You're certainly fronting the leader in testing on the oral microbiome. You've got a background in gut microbiome. Thank goodness you switched to oral microbiome, sake of all of us. And you've got a PhD, you've done research, you've helped write 11 peer-reviewed publications, I think 6 as the first author, and you are working on more. I know you are. You're a busy guy. Thanks for joining us today.
Absolutely. Thanks for having me. It's always my pleasure.
Thank you. So let's start off with just kind of a quick, what's new in the oral microbiome? What is the oral microbiome? And then, of course, we'll segue into why test for it. And then we'll really focus the rest of the hour on how to get the test. Certainly, that's easy. That takes minutes. How to take the test, that takes minutes. But what to do with the results. And we're actually going to go over some results. That's why I mentioned there's a video portion to this that would be interesting for people to see, especially for people that have already tested, certainly people that are going to test once they hear this. So let's start with, oral microbiome. What's the big deal? What is it? And why should we test it?
Yeah, so the oral microbiome is the community of bacteria, fungi, and viruses, of course, that live in your mouth. And at Bristle, we sample the salivary microbiome. So this is the bacteria and viruses and the fungi that are shed in your saliva. And the way we do this is we look at all the DNA from all the microbes there, so that we can tell what microbes are living in the spaces in your mouth. And why is this so important? Well, it turns out that these microbes are associated with not only oral health, so things like cavities and gum disease, both caused by bacteria, but also systemic health. So it turns out that cardiovascular disease, colon cancer, even Alzheimer's disease, there are potentially microbial components, and those microbes that live in your mouth actually have a substantial effect on progression of those diseases and your risk of those diseases.
Yeah, so and things are changing rapidly. I mean, I don't think we want to, think of the oral microbiome as just a small player. We know it's the second most diverse. It's linked with all the other biomes. Most of us understand that the gut microbiome is a big player, but the oral microbiome, there's a new study out, I think I shared it with you, on how as simple as an oral microbiome dysbiosis, which I would say is 50 to 60, maybe even 70% of us, especially after age 50, 55, we have an oral dysbiosis in our mouth. One of the manifestations of that is the #1 disease in the world, tooth decay. Periodontal disease is not that far behind. Huge cytokine inflammatory chronic autoimmune disease in the body that reaches out to all other parts of the body and affects it in many ways, is a causative factor in Alzheimer's, for example. So the study talks about, just to make it even more exciting for all of us, or dire, is that there's this link now to psychiatric disorders, even bipolar disease, correlating to an oral dysbiosis. So this bunch of bacteria and fungi and yeast cells in our mouth is pretty important. And of course, we've been trying to knock it down and disinfect it for over a century, actually longer. And thank goodness you've done research and supporting the fact that we need to nourish this oral microbiome. So obviously it's important. It's a big player in terms of our existence and our healthcare destiny, and even our mental makeup, our mental framework, and happiness, and all of that. So certainly a big deal. Okay, so let's talk about testing. Obviously, I think most people would know at this point why you would want to test that and quantify it. But what does that look like? What are we quantifying? And then, of course, we're going to get into what do we do with those results. But what actually are we measuring, and why are we measuring the correct thing, for example? I just want to be devil's advocate here. I mean, are we doing the right thing?
Yeah, really good question. So I'm going to take a step back and talk about older forms of testing to kind of give a framework of where we are today versus where we were maybe 30 years ago.
Perfect.
Okay, so way back when, actually probably 100 years ago, dentists and people knew that bacteria could cause gum disease and cavities. And the reason we knew this was because actually Anton van Leeuwenhoek, who invented the microscope, of course, was the first person who scraped someone's cavity, put it under a microscope and said, looks like there's worms in here. This person has worms that are digging into their teeth and are causing cavities. And of course, when they scraped healthy plaque, they didn't find any of those worms. They found a different morphology of bacteria, and they said, look, these are very different. It must be worms that are causing cavities. And since then, like, we've known there's a microbial component, but we could never see exactly what it was until recently because of the advent of molecular biology techniques such as sequencing. So probably 50 years ago, we first were able to really look at some of these microbes by actually doing culturing. So way back when, sometimes dentists or practitioners, they would take some infected tissue or swab an infected region and put it on a petri dish, and they would just see what would grow. And they would say, okay, because of the way that these things are growing, and if we do biochemical properties on them and we grow them in different types of media and see their behaviors, you can say, okay, this microbe here is probably Strep mutans, and that's why you have this cavity. And the way we can fix it is by getting rid of the Strep mutans. Or, in gum disease, similarly, you can say, okay, this person has a lot of Porphyromonas gingivalis, or this person has a lot of Tannerella forsythia. So we can use different antimicrobials based on what the microbes were. And of course, this was not a quantitative approach, because you're just using observational data. You're saying, okay, I saw something from a swab that I put on a petri dish and I figured out what it was. But fast forward now, maybe another 10 years, we started to understand that bacteria actually have DNA, all right? Bacteria and viruses and fungi, they all have nucleic acids in them. And this is the same across all life forms on Earth. It turns out that you can look at that nucleic acid. So for bacteria, you can look at the DNA and you can say, what type of microbe is this because of the DNA that it has, okay? So this is a very targeted approach where we can, instead of swabbing it and putting it on a plate, now we can take that swab and we can break open all the bacterial cells that are in there, and we get the DNA, and then we can use molecular approaches to say what's in there versus maybe what's not in there. And so this approach is really a detection versus non-detection. It's not very quantitative, but you don't need to do swabbing. You don't actually need to do observations. You can actually do a diagnostic approach to look for strep mutans, to look for PG, to look for different types of pathogens. Okay, and this is of course very targeted. In this diagnostic approach, you can't look at everything. You can only look at maybe a few dozen things at the very most. But back then, you would probably only do like 3 different things. And that approach was called quantitative PCR, which of course is still very relevant today, but it doesn't give you a very good picture of what's actually going on in the mouth because you're missing a lot of other information. You're only looking very targeted. It's like if you went out into the garden and you could only see orange, you can only count the oranges, but you know how many other trees there are. There's other apple trees and there's cherries, and you're completely missing the forest for the trees, basically. So fast forward a little bit farther, molecular biologists invented a new approach of looking at all the bacteria in a sample. And the way they did this is it turns out all bacteria have very conserved areas in their DNA. And they developed a technique to look at all these conserved areas and to determine what kinds of bacteria are in a community. Okay, so instead of a very targeted approach, now we're just looking kind of broadly, but only broadly at bacteria. Okay, so now instead of only having eyes that can see orange, now your eyes can see multiple different colors, and all of a sudden you can see the different trees that are in your garden. You can see all the apples. You can kind of count the ratio of the oranges to the apples. You can count the ratio of cherries to the rest of the things in the garden. But this approach is still missing a few things. The first is it doesn't actually tell you what type of orange. Of course, we have different types of oranges. We have a navel orange, or we have clementines, and we have sumo oranges. And this 16S approach doesn't allow you to look at what type of orange. It only says you have an orange. Okay, and so come to today at Bristle, we use what is the most up-to-date technology, I would say, is the highest resolution, most powerful technology we have, which is called Shaka metagenomics. And this looks at everything. So instead of just looking at whether or not you have oranges, apples, and cherries, we're looking at the parts of the apple. We're looking at the parts of the oranges and saying, how many seeds are in each of these oranges, and how many seeds are in each of these apples? What strain of apple is this? What flavor profile? We're looking at everything we possibly can to get as much information about all the things in your garden. So it's not just bacteria as well. In this technology, we can capture things like viruses. And we kind of have an idea of what they're doing because of their flavor profiles. We can see very deeply into how they behave. And so at Bristle, we use this technology because it is the one that gives the most information. And
so given that analogy, or maybe another analogy, what I like about where we're at with testing is that it is a positive identification of a specific species. It's like a thumbprint for a human being. We're all individual, and I know a thumbprint is not DNA, but it is a manifestation of DNA, and it gives positive ID. So that tells us exactly what is in our mouth. In terms of the biome. How do you measure quantity? Or each bug has a different colony count. How is that measured?
That's a really good question. So at Bristle, we use a thing called relative abundance. And what this means is that we're actually measuring parts of a pie. So if all the microbes in your mouth basically sum up 100% of a pie, and your mouth is the pie, we can say, hey, this much of your mouth is colonized by some species, and this amount of your mouth is colonized by orange trees, this type of orange. This part of your mouth is this type of apple. And the reason we do this is because there's actually very important interactions that happen between microbes that relative abundance gives a very good picture of. Okay, so you can imagine two different species that are, I'll call them antagonistic. Okay, so something like Streptococcus gordonii versus Strep mutans, right? If you measured the absolute abundance of both of these, okay, and let's say Strep mutans is at 5 and Strep gordonii is at 5, this is like total number of bacteria, exactly the number. If we measured those, we would say there's a 1-to-1 ratio, and you would say maybe you don't actually have an idea if this is dysbiotic or not, but if I tell you that this is dysbiotic, really you want a lot less Strep mutans, okay? The only reason we know that is because we're actually looking at the ratios of those bacteria, right? If I just look at Streptococcus mutans by itself and just say, you have 5 of these, that doesn't actually tell you the picture of there's Streptococcus gordonii in your mouth. Maybe you have 100 of it, and that's actually helping to prevent Strep mutans from causing damage to your enamel. You're completely missing that picture if you don't get a comprehensive view and you use, the pie as an example, where, the amount of sugar in the pie ratio to the amount of salt is actually very important for its composition.
Yeah, I mean, you're referring to modulation, and that's always been the case with the biomes. It's who's your neighbor? I mean, and what do they have on this bug? And so we're going to talk about that because that has a lot to do with how to interpret the results. One last question before we do that, David. You kind of nicely went through that timeline of, degree of resolution and over time. That was about a span of 200 years, I think. And it's funny, we've only really gotten really good at it in the last 20, 30 years. Is there, by chance, as a researcher, as a scientist, is there any degree of higher resolution that you're still wanting or needing or would like to see? And what does that look like? What is the next test?
That is
the degree of information that we get.
Fantastic question. I love this question. So there is one more deeper level. As I described, we can kind of get an idea of, the seeds and the flavor profile of each of the oranges and apples. Okay, but what if you could actually reconstruct all of them? What if you can say, okay, we don't just know the flavor profile, we also know exactly how you make this microbe, right? Make this orange from when it was a bud on the tree all the way until it became the orange. That would tell you a lot of information, right? And the only way we get there is by even deeper sequencing. So at Bristle, we use shotgun metagenomics, but we use very shallow level. And so the reason we do this is for cost, because it's very expensive to do deep sequencing, and we try to provide
time consuming. Yep.
Very time consuming. Yeah, and so we try to provide it to our customers at as low as a cost as is possible to get information, to get the minimum amount of information that is actually useful. But of course, if we got 10 times the amount of information, if we actually understood, all the functions of every microbe in your mouth, then we can get a much better picture of what they're really doing. Right now, sometimes we kind of have to guess. So just for instance, we may capture a piece of DNA from P. gingivalis that encodes one of its virulence factors. Okay.
Right.
And P. gingivalis may have 20 virulence factors, but we're only picking up one of them because we don't actually have a whole picture. We're only looking at like one of the seeds in P. gingivalis as an orange. So we can kind of infer, hey, maybe there are these other 19 there, but we're not really sure. And that's kind of the next level. It's like, we really know that you have these types of virulence factors in your mouth.
And I mean, just to be clear, because now that we're talking about not degree of accuracy, because the information you're getting is very accurate, but the degree of resolution, how many levels, how many layers of the onion are you peeling away? Bristle is right now on the market the most high-res, sophisticated, I don't want to say sophisticated, but the best test out there in terms of the information that you get, because it casts a wider net. There are some other tests out there. They've been in use for about, I want to say 10, 15 years, but they're only looking for 5 or 6 bugs, and they're only looking for bugs that would, like the strep mutant bugs or P. gingivalis bug, just the bugs that are the bad boys, and that really is a very dangerous way of making clinical decisions based on just a few players. And as you were inferring, the complexity of the oral microbiome, like virulence factors, if we were seeing one, but there are 18 others, 20 other virulence factors, it's all about how these bugs interconnect and how they control each other, modulate each other, boost each other, help each other, control their populations. And so we're still kind of at the beginning of understanding all of this, but thank goodness we've got Bristle. So let's now get into, let's spend the next half hour on, okay, so start with how to take the test. That's the easy part, and then we'll talk about the results.
Yeah, it's simple enough. You spit in a tube. So if you order your test online, we'll send you a kit where you can spit in a tube, send it back to the lab. A few weeks later, because we need to do all the processing, to do the deep sequencing, it takes time to get good resolution. You receive an email, and this email says, "Hey, your results are ready. Let's take a look."
That's pretty simple. Right. And this is, just to be clear, this is available to anyone. You don't have to order this test through a dentist or a physician. Certainly you can, but this is available to everyone. And I recommend, whether you want your dentist to order the test, because you can bring in the results, just order the test. And even if you think you're pretty healthy, get a baseline on, where you are in terms of your oral microbiome. There are a lot of factors that can affect oral microbiome within days. So anyway, go ahead, David.
I would love to talk about the changes, rapid changes in the oral microbiome. We have a lot of very compelling data about that, and
especially in kids.
Especially
kids are all over the map, and
put everything in their mouth.
One day we'll be testing our kids. I'm big fan of that, and I can't wait for that.
Yes. So at Bristle, what we try to do is boil down your entire microbiome, which is, could be hundreds of species in your mouth, into some easily digestible scores. So we give you a report card, and in this report card right now are 7 scores. There's potential to expand these later because of the data that we collect. I'm just going to talk a little bit about the scores individually, starting with the ones on the right, actually. So these are disease-related scores. So we have 4 disease-related scores today. The first one is halitosis. So halitosis is a very common issue. I think some estimates have up to 50 to 70% of people have halitosis and they don't know. It's not something that is traditionally, diagnosed by anyone in particular. You may go to your dentist and say, "Hey, I think I'm having trouble with some bad breath," and they may or may not help you. They may not know how to. So this is the halitosis score. So that's bad breath. The second score is gum inflammation. So of course, bacteria in your mouth, they can cause periodontal disease and gingivitis. And there are some that are much better at causing gum inflammation than others. And this score kind of summarizes that. The next is the tooth decay score. So of course, we all know that Streptococcus mutans, Strep sobrinus, Bifidobacterium dentium, Scardovia wixiae, these are all very important players in the progression of tooth decay. And people who have them are, at much higher risk of having tooth decay. And the last one that's disease-related score is a gut inflammation score. So you may know that bacteria in your mouth, they actually traverse through your stomach and they end up in your gut, and they can cause damage. They can cause havoc because they don't always belong there. And this is kind of a multifactorial inflammatory score, because people who have high gut inflammation scores, they may not have gut manifestations. And this is because their gut microbiome may be healthy enough to keep them at bay. But other people who have struggled for a long time with their gut health and have never been able to solve it, they may actually have an issue in their mouth where the bacteria in their mouth are just seeding constant inflammation in their gut. They can take millions of kombuchas a day and still have constant issues. And it's possible that it just starts in the mouth.
And tooth sensitivity.
And
from the acidic pH, right?
Yep. So these are our 4 disease-related scores. The other 3 are health-related scores. So these are positive effectors. So disease-related, of course, are kind of negative connotation. If the higher the score, the worse it is. On the opposite end, we have a commensal score. So commensal bacteria are generally thought of as beneficial bacteria that seed the mouth. And these are very important for a number of reasons. So they take up space so that other things can't grow into their space. They tune your immune system and dampen it. They help to reduce inflammation, so they can train your immune system to not be so reactive. They can also produce metabolites that help you do things like digest food. They can help produce vitamins, like even the microbes in your gut that produce vitamin K. It's very similar in the mouth. And of course, they can produce nitrate. They reduce nitrate into nitrite, nitric oxide, and this is a very important factor, which is also in the next score. So we have a nitrate reduction score, because we know now that the bacteria in the mouth, they perform a very important function here, and it's that they can actually produce nitric oxide.
So this is like the introductory page. When I look at these after my patients have taken the Bristle score, I would like to see all green. I don't see, the best score I've seen is all green, but typically it's all green with orange on the halitosis score. And as you said, it's pretty prevalent. It's no surprise to me, because that's for me as a clinician, it's an early marker for something is wrong with the oral microbiome.
Exactly.
And of course you can't, before testing, I would try and smell the patient's breath, or if the patient complained, or if a partner complained, that would lead me into the discussion of okay, well, we need to discuss and define for you the oral microbiome. So that's very helpful as a clinician. But if you have all green boxes and you're scoring high on halitosis, it doesn't mean you're healthy. It means that you're kind of teetering on the edge, and your oral microbiome is trying to modulate that part of it. So the halitosis score is important. That would lead me back to nitrate reduction. That is a score or a measure of having the right bugs. Typically it's everywhere, but on the back of your tongue, that are able to reduce the nitrates in vegetables. Again, eat the rainbow, so that you can produce this incredible gas that lowers your blood pressure. We have new studies out now that saying, well, we've had studies for a while now saying that mouthwash is killing the bacteria on the back of the tongue, which actually leads to elevated blood pressure. Now we have study, I think more than one, on just scraping your tongue can actually lower your blood pressure. So the nitrate reduction is very important. That's an important score. It usually ties in with the halitosis. That's where a lot of the halitosis occurs. When I see the commensal score, that's the first thing I look at. Just a large number of the good guys is great to see. That means that they're able to, keep down the numbers of the bad guys. Again, it's not this simple, but if that was a low score, then that typically is a very serious condition. Diversity, I think that is difficult as a clinician to really, and I know it is for you as well. And I've heard you say that too much diversity is bad as well. There's this sweet spot of numbers of bugs to each other. And that's why it's so important to measure every single organism in the mouth. And that's what Bristle does. And so the diversity score is tough, but if you have a low diversity score, that's not a good thing. And certainly
both ends of the spectrum are definitely not great.
Right. Yeah. And again, just to be clear, we're not going to talk about solutions in this 1 hour, because that would make it 2 hours, but that's probably the next podcast we'll do, right, David? We'll talk about, I just want people to, we want people to understand the test and to be impressed in the proper way by the results. And so anyway, tooth decay, that's an easy one. Most people look at that first. That's why I want to test kids, because I think the parents would really like to know, will their kid, do they have a predilection to getting decay, and can they do something early on before that happens? Because I've seen parents get the news that your kid has 5, 10 cavities, or even 1, and it's devastating. And so that it would be great to be able to give that information to a parent so that they can prevent it. And then gum inflammation, I can't wait till we get the Alzheimer's score. But for now, I use this as a way to motivate patients, but also to let them know that if that score was high, there is a worry and that patient is predisposed to Alzheimer's, and we can talk about that in the solution part two of this episode. But there's a lot of information just on the introductory page here,
Yeah.
and this information, I would argue, well, I mean, it's easy to argue because I know this because I'm a clinician. This is 90% more information than you're getting when you see a dentist on a regular basis. And it's quantified and it's scientific. And that's why it's so powerful. So whether your dentist recommends testing or not, just get this test, have this in your health folder, your dental health, oral health folder, and give it to your dentist. I think they would be very thankful that you've given this information to them. And if you have a high cavity score or gum inflammation score, then they know where to go with it. They can focus their protocol, their treatment plan on those items. So, but anyway, I know there's way more information, but I just think that one bit right there is amazing. It's a great start. It's just the introductory page.
Yep.
I know more is coming.
Yep.
You guys are working on more scores, and it's going to be, the nuances are coming, although this is already very nuanced.
Yeah. So let's actually dive into one of these scores.
Right.
So when you you can view your report card, and then each of these is interactive where you can click on it and jump straight to the page. So this is actually my test from 2021, and my wife complains that I have halitosis all the time, but my gums are perfectly healthy. Like, I go to the dentist and they're like, "There's nothing wrong with you."
Right.
Very annoying. But this objective test tells me, hey, turns out I do have disposition for halitosis. My halitosis score was an 8.5 out of 10, which is very high. So in our halitosis score in particular, it tells you kind of what bacteria may be contributing to your halitosis, and it gives you kind of a type. So at Bristle, we group them into different types. The three most prevalent ones are probably the gum liners. So there's bacteria that live in your gums but don't cause inflammation. So things like Dialister, and Selenomonas bacteria that can produce kind of volatile chemicals that smell foul. There's also the gum inflammatory species, of course. So usually, at least for most people, gum inflammation comes along with halitosis, and this is because the bacteria that cause gum inflammation, like P. gingivalis, Tannerella forsythia, or treponemas, they cause damage to your gums, and they're fermenting the things that they're causing damage to, and right. This is producing a foul smell. And the last one, or the next most common one, is tongue species. There are tongue coating species that we know live on the tongue, like Solobacterium, Atopobium, and these also produce foul smelling chemicals like hydrogen sulfide. And so those are the three most common groups. And my group is the gum liners because of the makeup of the bacteria that live in my mouth.
And a lot of that is location in the mouth. These little micro niches of the oral microbiome. And every week I find a new or read about a new micro niche. But yeah, certainly free margin bacteria are different than in the pocket, than in the vestibule, than in the back of the mouth or back of the tongue, as opposed to the front of the tongue. And probably based on saliva flow and exposure to oxygen or not. It again, very, very complicated ecosystem in the mouth. But go ahead, sorry.
It's an incredible ecosystem. And the way they all interact too, because saliva, of course, allows interchange between the different communities. And sometimes you can get invasion of some of the gum bacteria into your tongue. So a lot of people actually have a lot of fuso on their tongue. And they don't realize. That's just, and it becomes a reservoir for seeding constant fuso in other places in the body. Right. So these were the results from my test. So here you can see all the bacteria that you have. This
is the raw data.
This is the raw data. Yeah. So, and you can read about each of these bacteria. So like Tannerella forsythia, of course, it's a pathogen, and I have this in my mouth at the time of taking the test, and it's only present in about 42% of all microbiomes. And my score was moderate, and this is probably something I needed to work on. So the way to get rid of this, of course, is potentially to floss a little better. At the time, I probably wasn't using a water pick.
Tongue scraping, maybe.
And tongue scraping, of course, yeah, because a lot of these microbes. So like Atopobium parvulum is definitely one that lives on the tongue. And we're working on actually a little feature here that will describe a little better of how to get rid of them and what they're sensitive to. So certain microbes are more sensitive to certain probiotics or more sensitive to certain products.
Certain foods.
And certain foods. And this is a different conversation for probably another podcast, but that's the level of personalization that we can get to using this data.
Amazing.
So every score has something like this, which is a very detailed breakdown of all the bacteria that are related to this condition and the ones that are detected in your mouth. So just for instance, in my mouth for gum inflammatory species, they're pretty low levels. So the numbers here are out of 10. And usually we rank things from 0 to 10 based on their abundance in your mouth. And the way that score is derived is actually very interesting. It's actually based on a population level. So we actually compare your sample to all the other samples in the Bristle database, which is, I think, probably 1,000. It's probably approaching 10,000, maybe more than 10,000 now. And so that allows us to really determine what does your microbiome look like compared to all these other people who either have disease, don't have disease, who had disease at some point in the past and then had a surgery to fix it. We have a very large database that allows us to compare across different groups and what your makeup of your microbiome looks like compared to theirs. So you can see not only all the microbes that are in your mouth, but also the ones that were undetected. So we test for a lot of other things that you may not see, and you can see those all under each of these scores. And
that's not necessarily a good thing. Could be a bad thing because you want certain bacteria to help knock down some of the ones that you have levels that are too high. That's another thing that would be great to piece out eventually in these reports, like a recommendation like, okay, you've got an elevated bad bug, undetected good bug. This is what would happen. This is how your oral microbiome would improve if you were to elevate those populations of that bug, and then how to do it. So
that kind of gets into the commensal score. So in the commensal score in here, the undetected is very important because these are the ones you kind of want to boost. So like in my mouth, I had no Strep gordonii, and this is kind of important because Strep gordonii is really good at counteracting Strep mutans. Fortunately, I don't have any Strep mutans, but if it were to be introduced in my mouth, I would be more susceptible to it because I'm lacking this. So one of my goals could be to try and boost this. And it turns out that certain foods can help to increase this level, and certain probiotics even can help to boost this.
Exactly.
So that is a pretty good summary of all the information you get from just the results section. For people who are more inclined to look at their data in a way that is a little less structured so that you can explore it for your own, we actually do have this feature called the raw results table. And the raw results table allows you to do your own exploration.
Or your provider. I mean, if they have
anybody. This is my favorite feature, actually.
Of course it is. You're a researcher.
And this is how I consume the information.
Exactly.
So if you click on if you look at all the detected microbes in your mouth, that's what the raw table is. That's what this raw result is, and you can see everything. And you can sort them by the relative abundances. You can sort them by how high they are compared to everybody else. So just for instance, I have the highest abundance of Haemophilus sputorum than anybody else in our database. Well, of less than 1% of the people in our database. So I have a very high level of this. What does that mean for me? I don't really know yet because it's being investigated. Well, from this dashboard, you don't really know, but I personally know Haemophilus sputorum is actually a really good commensal bacteria.
It is.
That can do some really cool things. And it's part of the family of, it's part of the genus of bacteria that can reduce nitrates. And this allows you to kind of do your own digging and figure stuff out for your own.
Also, if you're a researcher, let's say you're testing products, you're a manufacturer, and you want to test and I think Bristle does do testing for people that are concerned about what is in their oral care
Yeah.
products and other things. You also, Bristle has come out with the only pre and probiotic that I recommend, their own brand of it, because you're looking at this degree of data, and
Yes.
there were a lot of bugs that were missing, and I'm sure that Bristle's going to branch out and probably have 10 different versions of this, because when you look at this data, you want to boost certain bugs that is you have to selectively feed them or knock them down. But it's a real puzzle that before you start unraveling it or applying solutions to the puzzle, you really need to have all the facts of what you're doing, because you can make things worse potentially, right?
Yep. Exactly.
Yep.
So there are actually, so on that point, there are actually a few gut probiotics that carry potentially cardiogenic species in them.
Mhm.
And so those are the things that we're trying to avoid. Like
Right.
we're trying to
Cardiogenic meaning cavity causing, just to be clear.
sorry, yes, I should.
There are some
I should have
products
added.
out there. Yes, right.
They produce acid from fermenting sugars in your mouth,
Mhm.
and they also happen to be able to colonize your tooth surface. So those
Right.
two activities together make them cardiogenic.
Right. Makes them potentially very pathogenic in terms of causing a cavity. And I mean, I have been asked by so many different manufacturers, and I've turned most of them down because I really didn't feel there was a good probiotic out there until now. And again, all of my decisions on products that I recommend have to be based on some study or form of or degree of scientific research. And that goes for toothpaste, it goes for so many different things. So this is another great aspect of oral microbiome testing, is now we can really re-examine all of our products, the ones that have been causing a lot of damage, most of them have, and then we can start formulating new ones that actually work and help and nourish the oral microbiome.
Yes, absolutely. And that's kind of the way that we approach building products too. It's we
Right.
look at all the data and we say, hey, like it turns out this thing is really beneficial for the vast majority of people, and that's the thing that we should try and make.
Right.
The place we haven't gotten to yet is really deep personalization though. Like as you can imagine, the probiotic that we made is probably for the is a general maintenance probiotic. So it's really good at helping most people because most people have an issue with gum inflammation bacteria.
Right.
But of course, it's lacking a lot of other things, and like
that's coming though. It's all coming, and
definitely coming.
just to be clear, this is the deepest amount of personalization I've seen so far with testing and also raw data. Do me a favor, click on, let's talk about gut inflammation. Look,
yeah, let's talk about gut inflammation.
Yeah, that's a really interesting one. And so Bristle is, of course, they're not measuring the gut flora, but you are able to make these, I don't want to use the word assumption, these conclusions indirectly, because you know about the link between bidirectional between the gut and the mouth. So talk a little bit about that. A lot of people think that everything in the mouth that gets swallowed, which is a lot, saliva, it's 1.5 liters a day, that it all gets destroyed by stomach acids. Talk about that a little bit. And obviously there is a link, and we know some of it does get past the stomach, and for good reason.
Yep. So I think the estimate now is that you swallow 100 billion bacteria every day. Because you're swallowing 1 liter of your saliva, and a lot of that saliva is carrying bacteria. And it depends, of course, the makeup of the bacteria that are getting into your stomach and potentially into your gut really depends on what's in your mouth, right?
Right.
So it turns out that after a lot of research, people have now found through very detailed experiments with not just human patients, but also in mouse models and in vitro models, that if you seed dysbiosis in the mouth with several different types of species, so things like Campylobacter or Klebsiella or Fuso or P. gingivalis, that those species actually end up in the gut. The way they get there is really confusing. There's two potential paths. The first is, of course, through the stomach, and they survive the stomach acid, and then they can survive the gut. And this, like the stomach acid doesn't kill 100% of all the bacteria. There's a fraction of them that will get through. And because you're swallowing 100 billion bacteria a day, even 1% is 1 billion individual
Right.
microbe that can
Bugs.
still end up in your gut. It's a
Individuals.
lot.
Yeah.
Individuals. And once those get through, if there's space for them in the gut, then they can colonize.
Right.
And the second potential path, which is really interesting, is that the bacteria that cause gum inflammation, they're actually being sampled by your immune system constantly. So there are dendritic cells and macrophages that are looking at what's in your mouth, and they grab those things, and sometimes they bring them back, and they say, hey, I need to figure out what this is. I'm going to bring it back to a lymph node. And so when it brings it back, actually some of those bacteria apparently survive. They survive that translocation. And as they survive, they can duplicate, and somehow they may end up in your gut. But one of the more compelling pieces of evidence that I've seen is that those bacteria, they don't even have to survive that translocation. What happens is the macrophage or the dendritic cell or the cell that is sampling this, the immune cell, it recognizes that this is an invader, and it becomes overreactive, and it actually trains the rest of the immune system to react to that same pathogen.
Right.
And it says, this Klebsiella is in a place it's not supposed to be. I need to attack this all the time.
Right.
It's almost like an autoimmunity. And now, because that Klebsiella has also ended up in your gut somehow by traversing the stomach, now every time you swallow and those Klebsiella end up in your gut, the same immune systems, your cells are now saying, hey, this shouldn't be here. I'm going to attack it all the time. I'm just going to attack.
Right.
And you end up with chronic inflammation.
Like in the joints, there's a oral bacterium for rheumatoid arthritis.
Very similar model.
And that's, you don't want to get there. And it's harder to reverse than it is certainly to prevent. And then would that, real quick, would that help? So oral microbiome testing will help when it comes to phage therapy, right?
Oh, absolutely. And that's my dream. My dream is the level of personalization down to phages, because it can be very personalized.
Yeah. Let's do an episode on that. I'm fascinated by that as well. Wouldn't that be fantastic? Because every person is different, especially when it comes to biomes. But that's incredible. So real quick, just to complete our discussion on the gut raw scores, give us an idea of what you'd like to see in that column.
I would like to see
lack of what if there's a lack of something, what would be a marker for concern?
Yeah, so this is actually a little bit of a concern. Mine is a 6.1. Actually, I used to have a lot of gut issues.
You're a new dad. You have the only excuse you ever need.
I'm a new dad.
Lack of sleep, poor diet.
So
come on.
My score is a little high. What I would like to see actually most people have a score that's below 3. It's like 3.3 to 0. And that's relatively healthy. That's like a normal microbiome. A lot of people are similar to mine, where they have a lot of Campylobacter or Fuso, and these can traverse the gut, and they can cause inflammation because they're in places they're not supposed to be. This microbe has a lot of information on it. Campylobacter concisus is because it is associated with IBD and diarrhea and Crohn's disease, and has a number of associations. And so it's this is one that I would like to see much lower, which I think I did lower in my next test.
Yeah, there's a connection between Crohn's and periodontal disease. I mean, it's amazing. I mean, the amount of information here, if you go through it as we did, I mean, we've spent at least a half hour on just these results. There's a lot of information here. And if it doesn't, I mean, it really, whether you're a practitioner or a patient, this literally should blow you away. This gives you so much information where you can actually do stuff to help yourself. And also it prevents you from doing the wrong things, or the advice that we've been given. It could be as simple as a mouthwash or an antibiotic. Timing is also crucial. When is the best time? What is the sequence of treatment? This information can be life-changing. Typically, I think it would be over the course of someone's lifetime, because so many people are getting the wrong treatment plan at the dental office. And again, it's not because we're trying to do the wrong thing. It's because we didn't ever have enough information.
Yeah.
But now we do. And so I think we're really kind of at the cusp. That's a bad word. I don't want to use that. We're at the crest of a breaking wave here. And finally, where we're seeing the possibilities of what we can do based on the level of information that we're getting, especially as dentists, but certainly as a layperson. This test is available to you right now. I mean, you could, my test would come in a few days. I would get my results in 2 weeks. That is an incredible amount of information, not just for oral health, but also for your overall health and well-being and even longevity. So anything else you want to discuss about data? Again, I agree with you. That is probably my favorite section as well. You can just peruse and then look up the bugs on the web, and
Yep, you can do a lot.
and then of course it could actually add to more confusion, because that's what raw data does. I mean, to
Yes.
collate all that data. So I would stick to the home page, the introductory page, start there. And if you're interested, this is wonderful information. What other scores are you working on that you can talk about?
Yeah.
What's coming? I mean, to Bristle.
Really good question. The score that I am most actively working on, which is not very active right now, because we have a lot of other features that we'd like to release to improve comprehension of results. But one of the most pressing things is probably thrush, and I would really like to see a fungal score because it's something that we have a lot of evidence that we can detect very well.
Right.
But we haven't done a lot of work on it lately, but that's
Right.
the next score that I would like to see. And there's also one more, which is not so common, but for the people who have the issue, it's a really big problem, and that is burning mouth syndrome.
Right.
I don't know if your listeners know, if anyone's unfamiliar with burning mouth syndrome, it's this very painful and itchy sensation of your gums that is
Right.
not caused by an etiology that's known.
Right.
But it turns out that for a lot of patients, that it is caused by a herpes virus. And that treatment with an antiviral for an extended period to get rid of it, because it takes a long time to get rid of herpes, actually treats them and fixes it for good. And if we can do that for patients, like, I would love that. I would love to help those people.
That would be wonderful. My patients are very uncomfortable about that, getting the diagnosis, but certainly living with it.
Yeah.
And it really kind of keeps them away from work and social events, and it's painful. It's extremely painful. And hopefully the antivirals get better. They are hard systemically, especially on the liver, but at least they're very selective. I don't know if you would agree with this, but from what I've read, they are selective enough where, because it is a long protocol, that you can give it to someone and not expect any big change to the bacterial numbers or components of the oral microbiome. But you're absolutely right. Oh, that would be wonderful. That would be fantastic. And a lot of dentists just recommend swishing with salt water and just, getting through it. But if they would understand that it is related to a specific, bad boy, bug, virus, then I think that would help them change their protocol and treatment plans, and it would make a lot of patients a lot happier. That's wonderful. Anything else?
Yeah, I'm just going to add, so we'll probably discuss this on another podcast, but something else that you always get from your results is a care plan. And a care plan is specifically tailored to you based on your results, and it has different recommendations that we make based on what we found in your microbiome. And we're not going to talk about them today, but the one thing I will say is, because this is a lot of information, it's a lot to understand and take in, and of course, as a layperson, it's really difficult for you to look up all the bugs and say what you should do next. We do have this very important feature, which is that you can talk to a health coach, an oral health coach who is well versed in the oral microbiome. And sometimes when you schedule this is actually with me, and sometimes when you schedule this with Eman or with one of our other talented coaches like Heather or Charity. But they are great, and they are a resource as well, on top of all the information that we provide. They have years of experience in dental hygiene, and now that they've been trained with the oral microbiome, and putting them together it's like magic.
Right.
So I would use that as well.
That is by far the best thing we've said in the last hour here. What a privilege to be able to talk with you or Iman. She's wonderful. And anyone that has knowledge of the oral microbiome, talk about customization and kind of just a fresh look. A lot of patients, I would say the majority of dental patients, because of the nature of how we treat them in our profession, unfortunately, are confused, as well as the practitioners. They're doing deep cleanings, scale and root cleanings. They're doing periodontal surgeries, and at best, we're trying to arrest a chronic disease, which has systemic implications. And I would say to anyone that sounds familiar to, or gets a lot of cavities every 6 months or even every year, get this test, get this degree of customization on coaching while it lasts, right? I mean, you know, as the numbers increase of people testing, this is going to be more difficult for you to provide.
There's already quite a long wait list. I think the earliest you can look out right now is like three weeks maybe. Yeah.
Right, which is totally worth it. And my wife, who is in healthcare, was in the healthcare field as well, and in biotech, she was very impressed with the coaching. And it really is an impressive feature that we don't talk enough about. But we are going to do that. We are going to talk about, we've talked about interpreting the results, how to get the test. That'll be part one. Part two, I'd love to spend an hour on all the things you can do. I'll get some questions together, perhaps maybe a specific case, an interesting case that we could all learn from. Okay. And then there's so many things that you can do, even without seeing your dentist. Certainly you would want to see your dentist. But it's important that what's happening, what's being done at the dentist is something that is based on this data. And otherwise you could just be spinning your wheels and your pocketbook. So David, this is incredible. This is one of the biggest shifts in dental care that I've seen in my 40 years of dentistry. And I would say it's the most significant. And I'm so pleased that this is now available to the profession. It's going to make us all better. It's going to make our patients healthier and happier. So whatever you're doing, just keep doing it. It's great stuff. And again, oral microbiome, just the advent of knowing and measuring it and being able to quantify it is going to change healthcare, I think, in beyond just oral health. I've spoken to a lot of influencers, Dr. Perlmutter, for example. He was very excited that this was available. He's on the cutting edge on research and comprehension of how bugs in the mouth can lead to Alzheimer's, for example, that can harm the brain. And this is stuff we can intervene at an early age. Also being able to, I say this often, but I only say it in the context of Bristle because it allows us as a patient to be aware of, and as a practitioner to actually do something about it, to treat in a prodromal phase of disease. In other words, no symptoms have appeared, but because we're able to test at this degree of resolution, and this is why you should test even if you think everything's fine. Even if your dentist thinks everything is fine, test, because things will pop up where if you treat in that symptomology stage of disease, it radically is different, cheaper, more effective, better outcome, and less painful in some cases. Yeah, and less exposure to pharmaceuticals and medications. Resetting the oral microbiome, a very dysbiotic microbiome, is a big deal, and it takes time, and it means using very strong bactericidal mouthwashes, and it doesn't always work. I mean, it's kind of hit and miss. Even though we have all this information, it's less hit and miss than it was before the days of testing. So again, what Bristle is doing is changing not just dental care, but healthcare overall, big picture. It's wonderful. Anything else you want to add, David? We're certainly going to talk hopefully soon about the next step. It's like, okay, hopefully we've explained well enough how to get the test, how to find the results, interpret them. What's the next stage? And we touched on it a little bit with the care plan, but there's lots to discuss. Anything else you want to add before we sign off?
Not really. Just, we believe a lot in prevention, and that's the goal of the test, is really to prevent the onset of any irreversible disease like a cavity,
Right.
painful and expensive.